Inside your body, 38 trillion bacteria are quietly deciding how healthy you actually feel today. They are not silent passengers sitting still. They react instantly to every single meal you eat. Every fast you attempt, every missed meal, every skipped breakfast sends them a direct chemical signal. But push that fasting window past 72 hours, and something extraordinary begins happening deep inside your intestines. Your cells start eating themselves in a process scientists call cellular self-cleaning, known as autophagy. What actually happens to the trillions of bacteria living inside you during that process? That question alone should completely change how you think about fasting forever. Your gut is not just a simple tube that digests food and disappears quietly. It is a living, breathing ecosystem, home to nearly 1,000 different bacterial species. Together, these bacteria carry genetic material that outnumbers your entire human genome by 150 times. Sit with that number for a second, because it completely reframes what your body actually is. You are not one organism. You are an entire, walking, breathing ecosystem of cooperation. And the lining of your gut happens to be one of the busiest surfaces in your entire body. Your intestinal wall rebuilds itself completely every 3 to 5 days without you noticing. The cells responsible for that rebuilding are often the very first ones to start autophagy. The moment nutrients disappear, these cells begin cleaning house before almost anywhere else in your body. Your gut is not simply hosting bacteria like a landlord ignoring its tenants completely. It is constantly negotiating with them, exchanging signals, chemicals, and survival strategies every single hour. These bacteria produce fatty acids, brain chemical precursors, and compounds that calm or trigger inflammation. When your eating pattern shifts dramatically, these microscopic factories immediately shift their entire output with it. Understanding this baseline is exactly what makes the 72-hour fast so fascinating. During the first 12 hours, your body is still comfortably burning stored sugar from glycogen, but inside your gut, something mechanical is already quietly changing without you feeling anything at all. A powerful cleaning wave called the migrating motor complex finally takes full, uninterrupted control. This wave usually gets paused every time food enters your digestive system throughout the day. Now, with no food arriving, it sweeps through your intestines completely undisturbed for hours. Bacteria that depend heavily on sugar and simple carbohydrates immediately start running low on fuel. Meanwhile, other bacterial species begin competing aggressively for whatever scraps remain inside your colon. Research published in Cell Host and Microbe found measurable bacterial shifts within just 10 hours. That means your gut ecosystem starts transforming long before your body even reaches ketosis. At the same time, your gut immune system quietly begins recalibrating itself in the background. Antibody levels lining your gut stay fairly stable during these very first hours of fasting, but the overall inflammatory tone inside your intestines has already started noticeably settling down. Scientists studying this exact relationship describe your gut as extremely sensitive to nutrient changes, and this sensitivity means the recalibration process is already measurable within the first 12 hours. Once you cross 16 hours without food, your glycogen reserves are seriously running low. Your liver responds by ramping up ketone production to keep your brain fully fueled. This single metabolic shift directly changes the chemical environment sitting inside your entire colon. As glucose disappears, the acidity levels inside parts of your gut begin slightly shifting. This new chemical environment starts favoring completely different bacterial populations than the ones before. This exact window is also when autophagy inside your gut lining becomes truly significant. A pathway called mTOR, which normally blocks cellular cleanup, finally starts losing control. Once that grip loosens, your cells begin forming tiny structures that hunt down damage. These structures swallow broken proteins, damaged compartments, and cellular debris piling up over time. Something remarkable is also happening along the mucus layer coating your entire intestinal wall. This mucus layer happens to be home to a bacteria called Akkermansia muciniphila. Researchers studying this species have linked it directly to gut wall strength and metabolism.
During fasting, this bacteria actually increases because it can survive by consuming mucus itself. Scientists are still debating whether this is a helpful adaptation or a genuine stress signal. Not every bacterial species inside your gut reacts to fasting the exact same way. Your 72-hour fast basically functions like a brutal survival test for microbes. Species that can adapt their diet tend to hold steady or even expand slightly. Species that only survive on constant food input start collapsing sharply within days. Certain carbohydrate-dependent bacteria show dramatic reductions once dietary fiber completely disappears from your gut. Meanwhile, bacteria capable of feeding off other bacteria's waste products actually start thriving instead. One powerful bacteria called Faecalibacterium prausnitzii produces butyrate, a crucial fuel for your colon cells. Its survival during fasting depends heavily on byproducts released by completely different bacterial species nearby. This creates a fascinating feedback loop between microbial cooperation and your intestinal cell survival. Meanwhile, Akkermansia continues expanding steadily, and researchers now suspect this expansion might actually help you. Instead of simply damaging your mucus barrier, it may be stimulating renewal and thickness. Studies have found that higher Akkermansia levels are linked to stronger, less permeable intestinal walls. This gives its fasting-related growth a surprisingly positive biological meaning that few people expect. As your fast crosses 24 hours and moves steadily toward 48 hours, everything intensifies. Autophagy inside your gut lining cells is now running at a dramatically elevated pace. Scientists emphasize measuring the complete cycle, not just the initial formation of cleanup structures. This complete cycle involves forming, merging, and successfully breaking down damaged material inside your cells. This process serves purposes far beyond basic cellular housekeeping happening quietly inside your intestines. Special cells called Paneth cells, located deep inside your intestinal crypts, depend heavily on autophagy. These cells release antimicrobial peptides that directly control which bacteria live near your gut wall. Research shows that when these cells lose proper autophagy function, microbial chaos and inflammation often follow. During extended fasting, autophagy activation inside these cells may function as strict quality control. It keeps antimicrobial output precise, even while your entire body is under serious metabolic stress. Your intestinal stem cells are also engaging autophagy in ways that boost their regenerative power. One landmark study found fasting actually enhances stem cell function through a specific fat-burning pathway. This enhancement appears directly linked to autophagy happening inside the stem cell environment itself. In other words, your gut's ability to renew itself may depend on this exact process. Your gut and your brain are in constant, non-stop communication through multiple biological channels simultaneously. During a 72-hour fast, this communication does not shut down or pause. Instead, it completely changes its language, tone, and the type of signals being sent.
The vagus nerve, which carries information from your gut straight to your brainstem, adjusts accordingly. As your intestinal movement patterns shift, the entire pattern of nerve signaling shifts right alongside it. One of the most powerful connections here involves specialized cells that sense your gut environment. These cells release hormones based entirely on what bacteria are producing at that exact moment. As fermentation activity changes during fasting, the hormonal signals reaching your brain change dramatically too. Researchers studying gut-brain communication have found that bacterial byproducts directly influence serotonin production levels. This matters enormously since roughly 90% of your body's serotonin is actually produced here. Tryptophan, an amino acid from food, gets converted differently depending on which bacteria are active. This balance determines whether tryptophan supports mood chemicals or takes a completely different metabolic pathway. During extended fasting, as dietary tryptophan disappears, this entire balance reorganizes itself in real time. Researchers have described this exact pathway as one of the most responsive systems during fasting. This finding adds a surprising neurological dimension to something that initially seemed purely digestive in nature. Chronic low-level gut inflammation remains one of the most misunderstood topics in modern medicine today. Your gut immune system stays in a constant state of controlled, careful activation at all times. It manages an incredibly delicate interface where trillions of bacteria live extremely close to your bloodstream. So what actually happens to that inflammatory tone after 72 hours without eating anything? It is not a simple switch flipping on or off inside your body overnight. Instead, it is a slow recalibration involving multiple overlapping systems moving at completely different speeds. One heavily studied pathway involves a signaling molecule that drives production of inflammatory chemicals throughout your body. Research shows that fasting suppresses this pathway, specifically inside your intestinal lining cells directly. Remarkably, this suppression happens through the exact same pathway responsible for triggering autophagy in cells. In other words, the same signal turning on cellular cleanup also turns down harmful inflammation simultaneously. These are not two separate, unrelated events happening by coincidence inside your body during fasting. They are deeply interconnected responses triggered by the exact same underlying nutritional state change. Butyrate deserves special attention here, since butyrate-producing bacteria remain active throughout your entire fast. They survive by feeding on byproducts from other bacteria rather than requiring direct dietary fiber. The butyrate they continue producing keeps reaching your colon cells and fighting inflammation effectively throughout. Research shows butyrate directly influences immune cells responsible for calming down excessive inflammatory responses naturally. This means your microbial contribution to immune regulation does not simply stop when eating stops. The idea that fasting completely resets your gut microbiome has spread widely across popular health media. But the honest scientific answer is far more nuanced and honestly far more interesting than that. Research does not show a complete replacement of your bacterial populations happening during a fast. Instead, it shows a temporary, partially reversible reorganization that depends heavily on what happens afterward. One major study examined gut microbiome changes during a month of daily intermittent religious fasting. Researchers found that microbial shifts during fasting were largely reversed once normal eating resumed again. However, the direction of that recovery depended heavily on the quality of food eaten afterward.
A separate study on extended water-only fasting found something particularly interesting worth mentioning here. While overall bacterial diversity temporarily dropped, certain key species showed changes that actually lasted much longer. One leading microbiome researcher has argued that your gut is far more dynamic than previously assumed. It can shift meaningfully within days, not months, which makes this 72-hour window genuinely significant. Whether this translates into permanent long-term change remains one of the biggest open questions today. When you finally reintroduce food after 72 hours, your gut does not simply resume normally. Instead, it enters a heightened state of responsiveness where what you eat first matters enormously. Researchers have specifically described this post-fast period as a window of elevated microbial flexibility. Your bacterial communities are far more open to change here than during your normal eating routine. Your intestinal lining also begins rapid structural recovery the moment nutrients start flowing back in. Proteins responsible for controlling intestinal permeability begin restoring their proper configuration almost immediately after refeeding. One study found that reintroducing specific fibers after fasting expanded certain beneficial bacteria before others recovered. This suggests the very first foods you eat shape your entire microbial recovery trajectory afterwards significantly. Meanwhile, autophagy itself gradually returns to normal baseline levels as nutrient signaling resumes throughout your body. But here is the fascinating part that most people completely overlook after fasting ends. The cellular improvements achieved during fasting, like clearing damaged components, do not simply disappear afterward. Research consistently shows that benefits from a completed autophagy cycle are not reversed simply by eating. Your gut lining after 72 hours of fasting and refeeding is measurably, physically different than before. The full scientific picture here is not about one single mechanism working alone in isolation. It is a story of deep interconnection between your microbiome, your immune system, and your brain. Nothing in this system exists independently, and that complexity is exactly what excites researchers studying it further. Scientists are now using fasting as a controlled window to study gut biology with remarkable precision. Removing food temporarily eliminates countless confounding variables that usually make this research incredibly difficult to isolate. This gives researchers an unusually clean signal for studying bacteria, cells, and cleanup processes directly. Multiple leading laboratories are now investigating how nutritional stress shapes long-term microbiome stability over time. What 72 hours of autophagy ultimately reveals is that your inner ecology is never static. It is constantly negotiating between bacterial demands, cellular maintenance, immune defense, and shifting metabolic states. Fasting simply compresses that entire negotiation into a window science can finally observe clearly. And what researchers are uncovering is not just about fasting as a simple health trend. They are quietly building an entirely new map of how your body maintains itself daily.